Excellent. So if, Hélio, if you'd like to get us going, that'd be great.
Yeah. First, say thank you so much to the Gorilla team for organizing this conference. I'm going to share a little bit about my experience transitioning from lab to online research. We do quite a bit of both, and I'll share this from the perspective of my PhD. So I'm doing my PhD in the clinical social cognition group here at Oxford. I'm working with Professor Geoff. Okay, so I'll try to do this presentation trying to detail how did we get to work with research online and specifically using Gorilla platforms. My PhD focuses mostly on investigating social cognition in autism and also how people process face dynamics during social cognition tasks. And I'll try to finish by sharing some lessons from conducting research online. So just to give you some background about this research, so most of you probably heard about autism in some shape or form, that it's characterized mostly with problems in social communication and potentially impaired empathy, emotional recognition, and reduced eye contact. So during my PhD, I'm actually working mostly on eye tracking in autism, and the goal is to try to build models that explain or that account for the heterogeneity in emotion processing abilities in autism. And this involves a lot of eye tracking research in the lab and also physiological research in the lab. And I'll start with this because this is actually a study that motivated a lot of the research that we are now conducting online on Gorilla platforms, not only because of COVID, but even before that, it was a good platform to allow us to address some of the limitations that we couldn't address in the lab. So we basically were running a study where we looked at how dynamic features in facial expression, so in real life, our faces move when we talk or when we express emotions, and we were trying to see how that dynamically shaped attention of participants, specifically participants with and without autism. And we try to build some models to try to explain how the dynamic allocation of attention shifted over time and so on. And what we found in that study was that a lot of the atypical gaze that people believe to be associated with autism, so that is reduced eye attention to eye region of others' faces, wasn't actually driven by autism. So this graph here, I'm showing how gaze changed over time in clinical and non-clinical groups. And we found that this condition called alexithymia, which I'm splitting here between high and low, seem to actually be explaining a lot of the variance in allocation of attention to the eye region over time. So alexithymia is this co-occurring condition that affects about 50% of people with autism. And it's quite frequent in autism, but it also happens in non-clinical samples. And it's characterized by difficulties with describing feelings in words, but also with difficulties describing physiological sensations. And we built a couple of different models accounting for autistic traits, anxiety, and other potential confounds. And we found that the alexithymia models were the ones that describe eye gaze behavior best. This was also the case if we looked at specific time scales. So for instance, here you're seeing the effect of alexithymia at different time points modeled by co-occurring autism alexithymia. And what this suggested is that this co-occurring condition might actually be really relevant to explain some of the social emotion processing deficits in autism. Of course, when you're studying comorbidity, which is typical in clinical condition, there's always the question on whether we are measuring the same thing twice. And this is certainly the case in the autistic research, where people question whether alexithymia is actually simply a symptom of autism. And one way to address this that we thought at the time was we could simply try to do a large-scale study and try to see whether we could tease apart or find an overlap between the constructs between alexithymia and autism. And this is actually what kickstarted a lot of our studies online. Basically, we took an approach of factor reduction. So we surveyed a lot of people through a combination of using Gorilla to host the tasks and Prolific to recruit participants. And we gathered about 1,000 people. And recently we also got a replication sample of about 500 people. And the idea here was that we could try to study overlap in the traditional factor structure sense. But a novel idea that occurred to us, and this has been emphasized by others, is this idea that psychopathology can be characterized as a network of related symptoms. So how related symptoms are can actually characterize a lot of psychiatric disorders. And that's exactly what we did from this sample. So the first thing you see here on top, it's basically a representation of factors extracted from autistic traits related questionnaires and alexithymia related questionnaires. So you don't need to necessarily know what's in, what are the specific items, but what I want to draw your attention here is that you end up seeing that we extracted about 11 factors, which if you know anything about the autism questionnaire and the alexithymia questionnaire, this is about the number that you would get if you sum the individual scales. And importantly, we have autism-related items factoring separately than the alexithymia-related items. And this was also illustrated when we did this by clustering using H clustering technique, where you see this area here clustering mostly items focusing on the autism component, whereas the bluish area captured items focus on alexithymia. And this was also the case if we took a more simple reduction technique such as PCA, which gave us more parsimonious components, but still separated clearly autism-focused items and alexithymia-related items. We then explored this network approach, basically to understand this overlap. So in a network approach, you could basically try to describe how related the items are using graph theory. And this is what we did here. And we're using multidimensional scaling, which renders the distances between those items interpretable as the Euclidean distances. And you see here the items on the left are mostly alexithymia items, and the items on the right are mostly autistic traits items, which supported this idea that although they co-occur, they do seem to be clustering separately. It seems that participants are responding differently for those items. Of course, some people may argue that it's important to check whether this is the case at the specific groups. And once we do that, you do see that overall, there's still a great split between alexithymia and autistic traits related items. But on the autism sample, what you see is that the symptoms are more connected, which could be predicted based on this network theory of psychopathology. So is it the case that we're measuring autism twice? I think this exercise showed us that this is not the case. And I think this was a good extension from a lab-based study where we were basically trying to split contributions of autism and alexithymia to actually try to investigate how this generalized when we try to study overlap. Just another example of how we approach this types of research that we usually do in the lab, like for eye tracking and psychophysiology, and how we move this online recently, is a study where we look at the contribution of different aspects of face dynamics to social processing, also in autism and alexithymia. So here's an example of typical data sets used in emotion perception research. So a data set which is of high intensity and low intensity. I actually realized that I flipped labels, so the bottom one should be less prototypical and this one is less prototypical. And we applied a common technique to animate faces, which works by linearizing transition steps. So this is a morph transitions. So basically this study was trying to suggest or to test whether the changes in dynamics that are introduced by stimuli data set types that are used in psychology, but also by techniques that we use to animate faces, can actually affect the cognitive processes such as perception of emotion, but also attention allocation during emotion processing. And what we see from a pure algorithmically driven perspective, so if we try to compute the amount of action coordination in the face is represent emotional expression. So this is basically action units, which are actions that are tied to specific emotions. You see that changes such as the linear animations of faces actually seem to affect greatly the time course of the expression, to the extent that in some cases, some artifacts are introduced when we apply such animations, where we see, for instance, action units that are not even present in realistic expressions, which suggests that this might plausibly affect behavior in emotion processing tasks. So we looked into this in a sample online as well. So we programmed a version of this task on Gorilla. And basically what we find is that there does seem to be an effect of the type of manipulation that we submit faces to, specifically into how they are rated, and also in terms of how that affects classification accuracy. So here I'm plotting the difference between accuracy from realistic faces and non-realistic faces, so the animated faces. And we see that from a scale from 0 to 100, here's a proportion from 1 to 0, you can have about a 20% effect of those manipulations. And this is particularly important when we're studying clinical groups. We also tested those samples online, where you see that although the general patterns for performance are consistent, so if people perform well in one task with realistic faces, they also tend to perform well in the animated morph tasks. But if you compute some sort of a representational similarity matrix for the performances, and which I transform this here into Fisher Z correlation, and you compare those within data sets and within groups, you see that performance for autistic participants here is actually more similar across tasks compared to performance for neurotypicals. So basically what we did here is that we computed those confusion matrices at an individual level to see whether those patterns of confusions were consistent across data sets. And in terms of our workflow, just to finish here, while we were running those studies, so this is the typical workflow that we use. I know a lot of people talked about this already, but just to give some idea of how we actually deployed it. So we usually do an initial pilot testing. So this is usually in the lab, and those projects also involve undergraduate students that are sometimes interning in the lab or doing their final year projects with us. And this initial pilot also goes to lab members, supervision, and this allows us to estimate, for instance, the duration. How long should we expect the test to last for people who are really good and people who perform more slowly. And then we move on to, even though we're doing this on the lab, we're still doing it on the Gorilla platform, because we want to have a sense of how people actually use this platform in the lab when we can actually observe them. And then we do an online piloting session. So this is with actual participants, and we usually enable a feedback for the initial batch of people that we have. And then we try to run this through our pipelines. We try to build the analysis pipelines to make sure that we're actually getting the data that it's analyzable as we intended. The third stage, which is still part of this iteration for task design, is a limited testing. We usually set between 10 to 20 people. It depends on the type of study. And then we deploy the study finally in platforms such as Prolific. We usually apply filters for language, ethnicity, depending on how broad we want to expand. But crucially, we usually have a support available during the testing hours. This is something that became apparent because participants sometimes have issues, and usually our undergraduate students in the lab basically stay during the times when the tasks are online. And we also integrate attention checks. So for instance, in the face tasks that I showed you there, we have attention checks that look more or less like the task. So basically they are presented with faces, but instead of the typical ratings for emotion processing that they had to do, they had to make an unrelated rating. So in this case, we had flashed a few words during the video for a very short second in a few frames. And this allowed us to compute some sort of accuracy. And we also do a lot of cross-checking for eligibility.
Hélio, I just want to move things on. Can you just, if you have a conclusion slide, that would be awesome. Otherwise, I think we need to move on.
Yes.
Thanks.
So this is pretty much the workflow that we use when we're doing online testing. We're also exploring some alternatives for eye tracking that some people talked about today, but not necessarily using online eye tracking, but exploring a few methods to estimate a gaze position in Gorilla using behavioral estimates, which would allow us to give us not only the focus of attention, but also what people are actually processing during those tasks. Thanks again, and just say thanks to our funders, my team, supervision, and all of the participants that took part in those studies.
All right, thank you very much, Hélio.